And then, like, come on the next day, they're different.
Mm-hmm.
Like, the editorial staff come in here and watch movies at night. I don't understand.
How do you want this?
Maybe on that side, I don't know, a little bit better. I mean, I'll be standing like right
I have a feeling
here.
we won't be able to see you either way because of the lights.
And I tend. I'll be like right here. Like
Oh.
half the people sitting on that side. No, it's not. I just got in here.
Oh,
Relax.
you know, Tim and Sue were already left. They're like, get this.
Can't be here.
Left away. Oh. Oh. I was texting them to come. So
That's for the best.
Well, this Stephanie's.
Just got in here.
Oh. Tim, I wasn't able to catch you with steps. Tried. And I stopped trying.
So one, she was supposed to like, way back, and then very end.
Really? I got jumped before?
No, I spent.
Oh, that's what I'm gonna do next year. Gonna wait to put stuff in at the last minute. Yeah, well. The accolades at the end. I'm not like the Golden State Warriors. They want the regular season title. No, and then we're not as energy in the postseason. I want to go look for the championship.
Working? Well, whatever. I just use my computer with this.
You going to the CMS workshop in Iowa?
I am not.
Are you? Yeah. I don't think you want to do that. Still been hopeful, but yeah,
The first thing this year is
that's right. I will. Right.
ensemble. Thank you very much.
So make a note of that.
So what is your pictures to
Starting video on this, looking at structural changes induced by good to regulate the
Again?
Hey, you want to sit on the end? Sit in the front?
It's good to see your family today.
Yeah, they were all there. I saw. I haven't got a chance to chat with you.
Tell me what everybody's doing online.
Neither do I. You know, he does try to explain, but she's doing well. I was just over to see my new grandson, who is, he was born 8 weeks early. So he's still in the hospital, but he's doing great. He just needs to grow. disappointment. So I sort of about every other day I run over there for a couple minutes and visit.
Yeah, that's much better than all that in such a short period of time.
Oh God, it was just so crazy. A couple weeks last time. But it was fun seeing everybody. Good dinners. Catch up with my sister. No, my mom is, but she's 89 and very frail. So she is not traveling. So she was sorry she couldn't come, but she's missed weddings and graduations and
Yeah, my mom's 61. Moving her back to the Midwest from North Carolina. Full operation. Time to do it.
well, we've been trying to get my mom to move closer to one of her daughters. We're, New York, North Carolina, Missouri, and she's in Florida. And because she's having none of it. She likes her house, and she's staying in her house. Doesn't matter. So she's a big girl. I can't make her do anything. So she fortunately is though, like getting help with stuff. She's got someone who comes to clean the house for her. She's talking about getting someone who can make meals. So that's good. Here's woman of the hour.
So you good now, Stephanie? Shall we? Are we good to go?
Oh, here.
Okay. All right, so I guess we'll get started. I'll make this brief. I don't want to take the spotlight away from the person of the hour today. So first of all, I'd like to welcome everyone to Stephanie's thesis defense, particularly members of her family and other friends and so on. Just by way of background, Stephanie hails from St. Louis, for those of you who don't know. She, I think, graduated from Lutheran North High School, is that correct? And then went to Valparaiso University in Indiana, where she was a biochemistry major. She was in the honors college and did very well there. And so we were more than happy to recruit her here for graduate school. She decided to pursue her PhD. She came here and has, as you'll see, she's done some really fantastic research and so on. And I'm not going to say anything about it other than to say that she's really taken something from ground zero and really turned it into a nice story. And for me, she's just really been a great person to have in the laboratory, really fun to interact with and so on. And her acerbic wit has really kept us all in line. So anyway, so without further ado, here's Stephanie.
Thank you, Ken, for that nice introduction, and thank you all for being here today. I'm excited to be able to share with you the work I've done in Ken's lab during my thesis research. It's been focusing on trying to elucidate novel functions for a classic negative regulator of G protein signaling in the nervous system. So in Ken's lab, there's been a long-standing interest to try to understand the mechanisms that regulate and control G protein signaling. And the reason that we're so interested in G protein signaling is because we know that these signaling cascades play a role in nearly every physiological process that you can think of. This is because the receptors that kick off these signaling cascades, or the GPCRs, make up the largest class of cell surface receptors in mammals. In fact, there are over 1,000 GPCRs encoded in mammalian genomes. And they control so many different physiological processes because they respond collectively to a wide array of ligands, including things like light and odors and neurotransmitters, chemokines, the list goes on. And so the way that these signaling cascades work in general is that when a GPCR is activated by a ligand, it activates a heterotrimeric G protein on the intracellular face of the plasma membrane. And so, as its name suggests, Heteromeroric G proteins are made of 3 components: an alpha subunit, beta subunit, and a gamma subunit. And when the alpha subunit is bound to GDP, this molecule is considered off and not in its signaling state. But when a GPCR is activated and it activates G-alpha, what happens is that the receptor promotes the exchange of GTP for GDP on the alpha subunit, and thereby activates the alpha subunit. Alpha and beta and gamma dissociate from each other. Then they go on to communicate to downstream effector proteins, eventually leading to the biological responses that are appropriate in response to whatever the original ligand was. Now, these signaling cascades are terminated when G-alpha proteins hydrolyze GTP back to GDP. And the alpha subunits have intrinsic GTPase activity. They can do this hydrolysis reaction on their own, but most of them are very poor GTPases. And that's where the RGS proteins come in. So RGS stands for regulator of G protein signaling. And what they do is that they bind selectively to activated G-alpha subunits and hydrolyze the GTPase reaction. And so for this reason, they're considered to be among the class of GAP proteins, which stands for GTPase activating protein. Now, it's the RGS proteins in particular that we in Ken's lab have been really interested in for many years now. And we've been so interested in these particular proteins because many studies over the years have shown that the mechanisms that negatively regulate G protein signaling are absolutely critical for normal physiology. So I think the easiest example of this to conceptualize is within your visual system. So phototransduction in your retina is controlled by a series of G protein signaling cascades. And if I were to walk, I'm not going to do this because the lights in here are a pain in the butt to fix, but if I walked over and started flipping the lights on and off, you would perceive those differences in illumination nearly immediately. And that in large part is due to the fact that the RGS proteins in your retina are faithfully terminating G protein signaling in the absence of photons. Now, if you didn't have any RGS proteins in your retina, it would take several seconds before you realize that the lights were turned off, which would pose a pretty serious problem for the usefulness of your visual system. This is just one example of a process where RGS proteins are absolutely critical, and there are many more in your body. And so since these proteins are so important, there's been a lot of research into them since they were first discovered in the early to mid-90s. And now we've come to appreciate that RGS proteins together actually constitute a superfamily of proteins that are divided into subfamilies. And the common feature of all these subfamilies is that they contain an RGS box. And this is the domain that's both necessary and sufficient to bind to active G-alpha subunits. And for those subfamilies that function as gaps, this is the domain that has that gap activity. This is what's necessary and sufficient to negatively regulate G-alpha subunits. Now, what I want you to appreciate from this slide is that the subfamilies of RGS proteins actually vary quite a bit in terms of their length and other domains that they contain. And research over the years has led us to appreciate that some RGS proteins can have functions other than their gap activity. So for instance, some members of the R12 family, actually all members of the R12 family, have an RGS domain that serves as a gap specifically for G-alpha IO subunits. But they also have a go local motif, and this is a motif that binds specifically to inactive G-alpha subunits and keeps them in the inactive state. And then they also have row binding domains that bind specifically to activated HRAS. So it's becoming appreciated that R12 proteins can function as integrators of both G protein signaling and RAS signaling. Another example of a multifunctional RGS protein are the GAFs. So GAFs bind to activated G-alpha 12-13 and inactivate them. But they also have a DH and a PH domain, which together bind to row A and activate row A. And this row A activation is actually in response to G-alpha 12-13 signaling. So within a single molecule, we have functionality that turns off G-alpha 12-13 signaling and also propagates signals downstream of G-alpha 12-13. Now, my project has been focused on the R7 RGS proteins. and trying to understand if this class of RGS proteins also has additional functions, just like the other classes that I've described. So let me tell you a little bit about these first. This class of proteins was first discovered through genetic screens performed in worms. And in these screens, they were looking for mutants that affected different neural behaviors, specifically feeding or egg laying. And time again, the R7 RGS proteins were the hits that came out of these screens. So upon further investigation, scientists realized that the R7 RGS proteins were expressed specifically in the motor neurons in the worm, and that they function as gaps for G-alpha subunits in the worm. Now, these early studies in worms actually taught us a couple of really important lessons. The first lesson was that R7 RGS proteins are absolutely critical for neuronal function, because when you either disrupt them, get rid of their function, or enhance their function, there were a vast or very apparent phenotypes on neurobiology in the worms. The other thing to appreciate from these studies is that there's something that was unique about the R7 RGS proteins that gave them their importance for neurobiology. And I say that because it was this class of proteins specifically that came out of these genetic screens. There were no other RGS proteins, there were no other gaps. It was this family specifically, suggesting that maybe it's not just their gap activity that's mediating these functions, there might be something else to them. So since these guys from the beginning seem so interesting, there have been several groups researching them over the past 20 years now. And we've learned a lot of lessons from understanding what these proteins do in mammalian systems. And the rest of my talk will be focused on R7 RGS proteins in mammals. So in mammals, there are 4 family members, RGS 6, 7, 9, and 11. And they function as gaps specifically for G-alpha IO. And collectively, they're expressed primarily in neurons and broadly throughout the nervous system. Now, the really striking thing about the R7 RGS family is it doesn't function as a monomer. It functions as a heterotrimer with 2 other proteins, G-beta5 and R7BP. Now, I'll talk more about these proteins in the coming slides. But before I get to that, I just want to point out the fact that these things function as heterotrimers is really striking, because like I said, we know that they're negative regulators for G-alpha IO, and it's the RGS domain of the R7 RGS protein that's responsible for that function, but they just look so much more complex than other known gaps for G-alpha IO. For instance, the R4 RGS family consists of basically just an RGS domain and a small tail, and this relatively simple domain structures is all that's really required to efficiently regulate G-alpha IO signaling, and to do this in a regulated manner. So just from the beginning, we were like, why are these things so complex? Are they doing more than just negatively regulating G-alpha IO? So let me talk about a little bit about the components of this heterotrimer. So like I said, R7 RGS proteins interact with G-beta5, which is shown here in cyan. So G-beta5 is a member of the G-beta subunit family. This is a member of the family that forms heterotrimeric G proteins and activates downstream signals in response to GPCR activation. The unique thing about G-beta5, though, is that it is never in complex with G-gamma or G-alpha in vivo. It only forms dimers or complexes with RGS proteins. Here is shown RGS9. And in fact, this interaction is absolutely obligate. If you get rid of G-beta5, you don't have any R7 RGS protein expression and vice versa. So they're always together. Now, excuse me, I have these crystal structures up here because I wanted to show that even though G-beta5 only interacts with R7 RGS proteins and not G-gamma subunits, the interaction with R7 RGS proteins does structurally resemble the interaction between a traditional beta and gamma. So here I'm showing G-beta5 in cyan, and then its primary interaction contact with the R7 RGS protein is with this green domain, which we call the Giggle domain. It stands for G protein gamma-like domain, or gamma-like, there's no T for domain. But what I want you to appreciate is this looks a lot like this, which is the structure of a traditional G-beta and G-gamma subunit. And the reason that this is interesting is because we know that these guys, upon G protein activation, dissociate from alpha and interact with and activate effector proteins. So the fact that these look so similar raises the possibility that maybe R7-RGS G-beta-5 complexes could also interact with effector proteins and activate them. Now the third component of this heterotrimer is R7BP, or R7 binding protein. This is a protein that was discovered in Kin's lab and another around 10 years ago now. And just like G-beta-5, R7BP is only found in complexes. It's never monomeric. So R7BP is only found in heterotrimers with RGS7 or R7-RGS proteins and G-beta-5. And for most of my talk, I'll refer to this complex as the R7 heterotrimer. Now, it's important to know that the opposite is not necessarily true. R7BP needs to bind to these for stability, but these don't always have to bind to R7BP for stability. So there are pools of dimer proteins within cells, but I'll mostly be focusing on these guys. So the best characterized function of R7BP is to serve as a membrane anchor for R7 heterotrimers. And that's because it has two lipidation sites at its C-terminus. And specifically, it's palmitoylated. And this palmitylation is actually really important. A lot of work by other people in our lab have looked at how and why R7BP is palmitoylated. And they found that since palmitylation is actually a reversible reaction, we found that in cells, palmitylation of R7BP is actually dynamic. And if you disrupt the cycle of palmitylation, if you disrupt either the adding or subtraction of palmitylation, R7 heterotrimers will accumulate in endomembranes in cells. And if you completely obliterate palmitylation on R7BP, the entire heterotrimer will accumulate in the nucleus of cells. So I want to come back to this idea that these things look more complicated than other gaps for G-alpha-IO. And since they just molecularly look more complicated, that raises the possibility that they might have other functions in cells. And moreover, they might have subcellular localization-specific functions, since the palmitylation state on R7BP can control where these guys are in the cells. Now, in addition to the fact that they look molecularly complicated, we also know that these heterotrimers are absolutely critical for normal neuronal function, because when we knock out all of them in the mouse, we get a variety of neurological phenotypes. I'm not going to walk through all of these, but I just want you to appreciate the breadth of these phenotypes. And since normally we do know that R7 heterotrimers negatively regulate G-alpha-IO, in the case where we get rid of them, we would expect that G-alpha-IO signaling would be up. And it stands to reason that some, if not all, of the phenotypes I had listed on the previous slide could arise because of an increase in G-alpha-IO activity. So one way to actually get at this experimentally is to look at the phenotypes of transgenic mice that express an RGS-insensitive version of the G-alpha subunit. So what these are G-alpha subunits that harbor a single point mutation that completely disrupts binding between them and an RGS protein, which renders them unable to be negatively regulated by RGS proteins. So the idea here is that if phenotypes in this mouse arise because of increased G-alpha-IO signaling, you'd expect similar phenotypes to be observed in this mouse. And for some of the R7-RGS knockout mouse phenotypes, that is true. So for instance, I just have one example up here. Both these mice are hyperactive. But this isn't true for all of the phenotypes that are observed in R7-RGS heterotrimer knockout animals. So for instance, in these knockout animals, the dendrites at the first synapse within your visual system are short and completely disorganized. But the dendrites, these same dendrites are normal in mice that have RGSI-insensitive G-alpha-O, and are also normal in G-alpha-O knockout mice, suggesting that there might be some other functionality of R7 heterotrimers that's giving rise to this dendritic phenotype. So this mouse phenotypic evidence, in combination with just the obvious molecular complexity of the R7 heterotrimers, led me to hypothesize that these heterotrimers likely have other functions, novel functions, in cells that are critical for neuronal biology. And if this is the case, then undoubtedly they have novel protein-protein interactions that mediate these functions. So the central goal of my thesis, or at least the beginning of my thesis, was to try to identify novel protein-protein interactions for R7 heterotrimers, then to subsequently understand what this means for the function of a neuronal cell. So before I took on this sort of fishing expedition to look for novel protein-protein interactions, I wanted to see if I could accumulate any sort of biochemical evidence suggesting that R7 heterotrimers do interact with other proteins within the cell. And so to do that, I performed chemical cross-linking experiments in a mouse neuroblastoma cell line. So this cell line expresses endogenous G-beta5 and endogenous RGS7, and is stably expressing a flag-tagged version of R7BP. So the principle here is that these cells are treated with DSS, which is cell permeable, and DSS forms covalent cross-links between the proteins that are close together. So essentially, DSS serve to covalently link the R7 heterotrimers with any proteins they might be interacting with at that time. Then I lysed these cells, isolated these complexes with a flag IP, ran them out on SDS page gel, and blotted for the R7 heterotrimer components. So here I'm showing you results from one of these experiments. This is a G-beta5 blot of the flag eluate. What I saw time and time again is that there were bands, distinct bands that were of apparent molecular weights that were larger than the heterotrimer. And this result is consistent with a model where R7 heterotrimers interact with other proteins within the cell. So with this preliminary evidence, or additional preliminary evidence in hand, we wanted to try to identify what some of these other proteins might be that are interacting with R7 heterotrimers, specifically in brain. So to do that, we generated a transgenic mouse that expressed affinity tag version of R7BP. So this is R7BP that has two small peptide tags that then served. So then this protein ended up serving as a bait protein for tandem affinity purification of R7 heterotrimers from brain. And specifically, the way we made this mouse is we targeted the tagged R7BP to the HPRT locus on the X chromosome of mice. This is a pretty standard, broadly used targeting strategy. And to make sure R7BP was expressed in neurons where it's normally expressed, we put the transgene under the control of the mouse prion promoter. So here below, I'm showing Western blots from a positive transgenic mouse and a negative transgenic mouse. You can see that the SFR transgene is expressed in various brain regions and absent from other tissues as expected, expressed to a comparable or slight level than endogenous R7. So then I use this protein in the brain as a bait protein to isolate R7 heterotrimers and any other things they might be interacting with. And the actual isolation process, which is called TAP, a lot of work went into optimizing this to maximize the recovery of my bait protein and to minimize the nonspecific background, and that's all summarized here. So on the left, I'm showing a protein-stained SDS page gel of final TAP eluate, showing that I get lots of proteins that pull down with my bait protein, but the nonspecific background was relatively low. So that's good. Right. Our Western blots for the various stages through the purification, I recover my bait protein as expected, and importantly, I show that I recovered G-beta5 and RGS7 my eluate. This is important because it showed that my purification conditions were appropriate to maintain heterotrimers together throughout the purification process, and then suggested that I was also potentially maintaining other protein-protein interactions, which is what I really wanted to identify. So then the real point of this was to figure out what other proteins co-purified with R7 heterotrimers from mouse brain. And so to do that, did the TAP purification, ran out my eluate, and then we excised gel slices, digested them, and sequenced the peptide by tandem mass spectrometry, and then identified what proteins these peptides originally came from. And I got quite a few results, but for the purpose of this talk, I've narrowed it to what I really want to talk about. And just a note first, for people that aren't used to looking at mass spec proteomics data, it's a sort of a standard bar in the field to in order to consider a protein as a positive hit in proteomics data, it needs to have at least 2 exclusive unique peptides from the mass spec data. What that means is these are 2 peptides that are absolutely only found in this protein being identified, and the peptides don't at all overlap. Okay, so from the mass spec data, of course, we identified R7BP, the bait protein, and several members of the R7 RGS family, and G-beta-5. We also identified G-alpha-O. So this is the G-alpha subunit that's known to interact with R7 heterotrimers, because this is a subunit that these guys act as gaps for. And intriguingly, we saw that the R7 heterotrimers also co-purified with G-alpha-13. This is another G-alpha subunit in a different family. And this stood out as interesting to us for several reasons. First, just from a biology background, we know that R7 heterotrimers are only gaps for G-alpha-I and G-alpha-O. They have no gap activity for any other G-alpha protein. So this was the first indication that R7 heterotrimers might be interacting with other G-alpha subunits via a relationship that's different from its relationship as a gap with G-alpha-IO. Additionally, I'd like to point out that the fact that we identified G-alpha-13 here, this was actually a very confident identification from the mass spec. Because like I said, there were 2 exclusive unique peptides, which is a bit of a feat for G-alpha subunits because they do share a decent amount of homology across everything. So these are 2 peptides that are absolutely only found in G-alpha-13. They're not even found in G-alpha-12, which is another family member of this family that has like 60% identity between the 2 for amino acids. So that was intriguing. And another point is that G-alpha-O is known to interact with R7 heterotrimers, and G-alpha-O is a very highly expressed protein in the brain, and G-alpha-13 is a very low expressed protein in the brain. So the fact that we were pulling this out was not just because it's all over the place and just an easy thing to isolate. So because of these reasons, we decided to follow up on G-alpha-13 on this interaction and try to understand what it means. And even though the mass spec data was very convincing, with any proteomics data, you need to have a separate method to confirm the interaction. And so to do this, I turned to split luciferase complementation. This is a pretty straightforward technique. Many of you are probably familiar with it, but just as a short explanation, the idea here is you take a luciferase enzyme. In my case, it was click-beetle green. You split that enzyme into 2 non-functional halves, shown here, and you put those non-functional halves on 2 proteins of interest. So here I'm showing a G-alpha subunit and an RGS subunit. And the idea is that if your 2 proteins, if you put these fusion proteins in a cell, and if your 2 proteins of interest interact, they'll bring the 2 halves of luciferase close enough together to refold together, reconstitute an active enzyme, such that when you add luciferin to the cells, this enzyme will produce light. So then light is the readout of the 2 proteins interacting. Before I actually went on to ask the question I was actually interested in, which is, can I confirm an interaction between R7 heterotrimers and G-alpha-13, I first wanted to assess the usefulness of this assay setup as an assay to detect protein-protein interactions for G-alpha subunits. So it had actually never been published before. So to do that, I did some proof of concept experiments, specifically looking at the interaction between 2 different G-alpha subunits and their known RGS domains. So here I'm showing that I get a nice split luciferase signal between a constitutively active version of G-alpha-13 and its known RGS domain. This is specific because I see no interaction between G-alpha-13 and an RGS domain for another G-alpha subunit, G-alpha-Q. And it's also dependent on the activity state because I don't see much of an interaction between wild-type G-alpha-13, which is predominantly inactive in these cells, and the RGS protein. And the same is true for the other G-alpha subunit I tested. So nice interaction between constitutively active GQ and its RGS protein, no interaction between constitutively active GQ and G13's RGS, and little interaction between G-alpha or inactivated G-alpha-Q and its RGS protein. So this assay from these experiments seemed to be appropriate to detect protein-protein or predicted protein-protein interactions for G-alpha subunits that was also sensitive to the activity state, the alpha. So then I moved on to the question I was really interested in, which is if I could confirm an interaction between G-alpha-13 and R7 heterotrimers. I was also, and if I could, I was also curious to see if this interaction had any specificity for a particular R7 RGS family member, or if it occurred broadly for the whole family, and also to see if R7BP was required to observe the interaction. And so what I found is that I could confirm an interaction between R7 heterotrimers and G-alpha-13, and this interaction occurred for every member of the R7 RGS family, and moreover, the interaction in every case was dependent on the presence of R7BP. And this result was specific for G-alpha-13 because I didn't see any interaction between G-alpha-Q and either RGS7 G-beta-5 dimers or RGS7 G-beta-5 R7BP heterotrimers, while I know that the GQ was still working because it did show a nice signal between it and its known RGS protein. So at this point, I had purified G-alpha-13 with R7 heterotrimers from brain and had confirmed this interaction with a separate method and had realized that this interaction seemed to be something that was broadly applicable for the whole R7 RGS family and also required R7BP. So then the real question is, what does this mean for cell biology, for neuron biology? And so to address that, took a step back and we're like, what do we know about the functions of these proteins already? So we knew, like I told you already, that R7 heterotrimers in cells function as negative regulators for G-alpha-IO. What we also knew was that active G-alpha-13 activates a ROGAF, which in turn activates ROA, which kicks off a variety of downstream signaling events that eventually lead to things like transcriptional regulation by the serum response factor and actomyosin or structure assembly and contraction. So then really the question is, oh no, this was happening yesterday. Okay, so I'll bring this back up. But really the question was, sorry guys, this is what happens when you demand too much from your computer over 6 years. So then the real question was, does the interaction between R7 heterotrimers and G-alpha-13 actually mean anything for the known functions of these proteins? Okay, hopefully that only happens once, but no way to know, it'll be a fun game. Anyways, right, so these guys interacting could have any number of functions on the, or any number of effects on the known functions of these proteins already. And I could have studied either arm of this diagram. Since going into this project, I was primarily interested to see if I could find any novel functions for R7 heterotrimers. I decided to focus my research on whether or not R7 heterotrimers affect known G-alpha-13 function. And specifically, I wanted to know if they affected G-alpha-13 function in a neuron cell context. So in neurons, when you activate G-alpha-13 via, for instance, LPA receptors, you get activated ROA, which manifests in neurite retraction in the neurons. So here I'm showing data from Stefan Offerman's lab, where where these are plated Purkinje neurons from either wild-type animals or G-alpha 13/12, which is a very close family member, as I already mentioned, knockout animals. And so when you treat wild-type animals with agonists, they retract their neurites, shown here. But when you treat these knockout neurons with agonists, there's no neurite retraction, which is shown here. So I wanted to use this cell biological readout as a way to assess if R7 heterotrimers at all affect G-alpha 13 function. But I really wanted to do this in a cell system that was slightly easier to manipulate than primary cultured neurons. So I went back to the neuroblastoma cell line I used for the cross-linking studies because these cells are super easy to grow, they grow fast, and you can differentiate them to look like neurons, and they're also very easy to transfect, which makes them easy to use for sort of these initial pass experiments. But first I needed to show that LPA actually does cause these cells to round up, because that had never been published before. And it does. So here is a differentiated into a cell treated with vehicle, and then when you add a maximum dose of LPA, the cells are very round. That's quantitated over here. So the way I quantitate all this data is I score a large number of cells for whether or not they have projections, and then it's expressed here as percent total cells that hit projections. So if the bars go down, that means the cells are getting more round. And then since LPA receptors actually activate a variety of G proteins, I wanted to confirm that this process I was observing was actually G-alpha 13 mediated. So to do that, I overexpressed the RGS domain that's specific for G-alpha 13. So I essentially overexpressed a protein that's known to turn off G-alpha 13 and inhibit it. And when I did that, the cells no longer retracted their neurites in response to LPA, shown here, or quantified, shown here. So with this data in hand, it told me that the N2A cells could, in fact, retract their neurites in response to LPA activation in a fashion that seemed to be dependent on G-alpha 13 activity. So then I went on to set up assay systems to actually ask the actual question I was interested in, which is, do R7 heterotrimers affect neurite retraction in these cells? And another reason that the N2A cells are actually a convenient model to use for this is because they express endogenous RGS7 and G-beta 5, but they lack R7BP. And since my split loop data showed that R7BP was required to see an interaction between the heterotrimers and G-alpha 13, I can make a stable cell line expressing R7BP. The idea is that in this cell line, we can get interaction between R7 heterotrimers and G-alpha 13. And you'd expect that to be absent in the wild-type N2A cells, control cells. So I took these cell populations, differentiated them, treated them with LPA, and then fixed and stained and scored them, just like I explained on the previous slide. And what I saw is that N2A cells, again, rounded up in response to increasing doses of LPA, which is quantified here. And the R7BP cells also rounded up with increasing doses of LPA. And their actual LPA response was really almost identical. That is, the change, the effect over basal at each LPA concentration was the same for both cell types. Instead, what was strikingly different is that the R7BP cells actually always started out ounder than the N2A cells. Their basal state was more round than the N2A cells. And so then the question became, why are these cells more round under basal conditions? We knew that LPA treatment was a G13 process and rounded up cells, but it really was unclear to us, or we had no way of knowing what sort of mechanisms were at play that were causing the cells to be basally more round. But we knew that there were a couple of reasonable options that I could test. So just in normal N2A cells, like I've already shown you, we know that if we have active G13 present, it causes neurites to retract. Another thing that's been published, though, is that if you activate G-alpha IO in these cells, it causes neurites to grow. So it's completely feasible that in the R7BP stable cells, when we have R7 heterotrimers at the plasma membrane, they could interact with G-alpha 13 under basal conditions and enhance the basal signaling or basal function of G-alpha 13, thereby increasing neurite retraction and leading to rounder cells. But another possibility is that if there's any level of G-alpha IO activity under these basal conditions, R7 heterotrimers could just be inhibiting this activity, thereby decreasing neurite growth, which would also lead to a net rounder phenotype in these cells. And really, I wanted to see if either one of these mechanisms were true here, or if, for instance, they were both true, because that was also a possibility. So to test the G13 arm, if R7 heterotrimers, if R7BP stable cells are more round because R7 heterotrimers are increasing G13 activity, then I would expect if I block G-alpha 13 activity under basal conditions in these cells, the cells should form neurites, much like N2A cells do. So to do that, again, I overexpress the negative or the inhibitor of G-alpha 13. And I saw that when this inhibitor was present, the cells went from a more rounded phenotype to one that bore neurites. And that is quantified here. And I just want to point out, this is data I had a few slides ago, that when I inhibit G-alpha 13 under basal conditions just in the wild type N2A cells, there's not much change in the morphology. So this data taken together supports a model where when R7BP is present, G-alpha 13 function is up, and that leads to the cells being more round. Now to test the other arm of these possible mechanisms, if R7BP cells, or if in R7BP cells, R7 heterotrimers are inhibiting G-alpha IO, and that's also contributing to the rounded phenotype, then I would expect that if I took the wild type N2A cells and inhibited G-alpha IO via another mechanisms, for instance by pertussis toxin treatment, then that would lead to a similar morphological phenotype as in the R7BP cells. So these cells would look similar. But when I treated the wild type N2A cells with pertussis toxin, I saw that they were able to have bear neurites just fine. They weren't any more round than in the absence of pertussis toxin. In fact, they might be even a little less round. This is the opposite from what I saw in the R7BP cells. So when R7BP is present, the cells are more round, but when GIO is inhibited, the cells are not more round. So the evidence in these past two slides, again, still supports a model where when R7 heterotrimers are present in these cells, they make the cells more round, and that seems to be due to an increase in G-alpha 13 activity under basal conditions. So to summarize the things that I found in my work, I first found that G-alpha 13 is a novel binding partner for R7 heterotrimers from brain, or in brain. And I also found that R7BP is absolutely required to observe this interaction, and that the interaction can occur with R7 heterotrimers containing any of the R7 RGS family. Now, intriguingly, I saw that R7 heterotrimers don't seem to affect G-alpha 13 downstream of activation of an LPA receptor. But via some mechanism that I still need to figure out, the R7 heterotrimers under basal conditions can enhance G-alpha 13 signaling and lead to neurite retraction, causing the cells to be more round. Now, these findings are significant in part because this is the very first time that it's ever been shown that R7 heterotrimers can interact with another G-alpha subunit aside from the interaction with G-alpha IO that's mediated by the RGS domain. This is, in addition, interesting because the interaction with G-alpha 13 seems to increase its signaling versus decrease its signaling. So I think in the future it'll be interesting to see if maybe these heterotrimers serve as a point of crosstalk between different G protein signaling cascades and neurons. And since this is just the very tip of the iceberg, the beginning of this study of what R7 heterotrimers mean for G13 biology, there are quite a few questions that still need to be answered. And some of them are relatively straightforward questions that can be answered soon with regards to sort of structure function questions. One of which is, why is R7BP required to observe this interaction? The easiest hypothesis is that R7BP interacts directly with G-alpha 13, and that's why it needs to be there. But another possibility is maybe RGS7 or G-beta 5 interacts with G13, but only when R7BP puts that complex on the membrane. Or for instance, only when R7BP binds that complex and maybe changes the conformation of it to reveal a G13 binding site. These are things that we have tools to really address in the near future. I think the most pertinent question to be addressed in the relatively immediate future is what mechanisms are at play that allow R7 heterotrimers to enhance G-alpha-13 function under the basal condition, and how is that different from how G-alpha-13 signals in response to LPA? So there are a variety of possibilities here as well. One possibility is maybe R7 heterotrimers interact with G-alpha-13 under basal conditions and somehow enhance spontaneous nucleotide exchange on the alpha subunit, thereby allowing the alpha subunit to become activated even independent from receptor activation. Now, ultimately, the most important question to be answered is, what does this interaction mean for neuron or for brain biology or development? And admittedly, I think we have quite a few questions that need to be answered just on the molecular basis and in cell culture before we can really design telling experiments in vivo. But I think it's both intriguing and also really important to at least speculate or have an idea in mind of what this could, or how this could inform our understanding of biology in vivo. So because I think it's important, I'm just going to spend a little bit of time at the end here talking about it. So one thing that we've known for some time now is that R7 heterotrimers are developmentally regulated. So here I'm showing Western blots from brain homogenates from mice at different developmental stages. What you can see is that both G-beta 5 and R7BP are dramatically upregulated during postnatal brain development, starting around birth, and then reach near adult levels at postnatal day 16. And we know that these heterotrimers are important for brain development, specifically for postnatal brain development, but the molecular mechanisms that are at play here, why they actually need to come on for the cells to function is unknown. And like I said, we know that these heterotrimers are important for postnatal development because when we get rid of all the R7 heterotrimers, the mice have a variety of phenotypes during postnatal development, including delayed maturation of cells in the cerebellum. They have delayed morphological development of the hippocampus, so the hippocampus changes shape during postnatal development. Also in the hippocampus, when you get rid of R7 heterotrimers, there are more ectopic cells. So these are cells that are in the wrong place in the hippocampus. And additionally, these mice have a variety of neurobehavioral phenotypes, including that they have either delayed or completely absent surface writing reflex. So you put a wild type mouse pup on its back, it wants to flip over. When you put these mice on their backs, a lot of them just lay there and don't really care. Now we don't actually know at this point what phenotypes arise from ablating G-alpha-13 activity during postnatal development, because those mouse models just don't exist yet. But I think everyone in the field would agree that G-alpha-13 signaling is undoubtedly very important during this stage of postnatal development, because it's very important during earlier stages of development as well. So for instance, when G-alpha-13 and its close family member, G-alpha-12, are knocked out specifically in the brain during earlier developmental stages, there are gross morphological defects in the cerebral cortex and the cerebellum, which primarily arise because certain neurons are over-migrating. There's an increase in ectopic neurons in these brain regions. So because of that, G-alpha-12 and 13 have been thought to provide some sort of stop signal for neuron migration during development. And so it's possible that they're also providing stop signals in the hippocampus during postnatal development, and R7 heterotrimers come on to enhance that function. And therefore, when R7 heterotrimers are absent, there are more ectopic cells in the hippocampus. Obviously, that needs to be tested, but it's just one thing to think about. Another thing I want to point out is one of the major biological processes that happens during postnatal development is synapse maturation. And so with synapse maturation, this involves pretty large rearrangements of the actin cytoskeleton to cause different shapes of dendritic spines. And there's also quite a lot of elimination of immature synapses. And so both of these processes involve a large rearrangements, as I said, of the actin cytoskeleton in a regulated manner. Since we know that G-alpha-13 is a regulator of the actin cytoskeleton, it's intriguing to hypothesize that maybe R7 heterotrimers come on during this time period to modulate G-alpha-13 activity during the process of synapse maturation. Now, as I said, all these ideas are really just completely speculative at this point, but I think it's exciting to think about how the basic research I did during my time here as a graduate student could one day lead to a deeper understanding of how G protein signaling and the regulation of G protein signaling informs both brain development and just adult brain function. And so now I would like to thank the people that were instrumental in the success of this project. First, I have to thank Ken. He's been a fantastic mentor. He's really mentoring both students and postdocs has always been a high priority of his, and he strikes a really nice balance between making sure we're on the right track and, working hard and then just making sure we can figure things out on our own. And I think that's really benefited me in my development as a scientist. I'd also like to thank the rest of the members of the Bloomer Lab, both present and past. You guys have all had very interesting points of view on my project. We work on a wide variety of projects, so it's been really nice to have fresh perspectives on things during lab meetings. And also they've been a lot of fun to work with. I want to give a special shout out to Kevin for, Michi and I call him our lab dad, mostly behind his back, but it's endearing. Anyways, Kevin, aside from the fact of making sure the lab doesn't just implode on itself on a daily basis, actually he generated and bred and maintained the mouse I used for this study, which is absolutely critical for its success. So for that, I'm very thankful. I'd like to thank my thesis committee for diligently giving me constructive criticism on this project throughout the whole course of the project, and also offering suggestions, all of which I'm sure you will continue to do very shortly. And I'd also like to recognize the proteomics core at the Donald Danforth Plant Sciences Center. They did all the mass spec sample prep and the actual mass spec for me. Particularly Ellen Marsh and Sophie Alvarez were especially helpful, and I was in constant contact with them during that stage of my research. But I'd also sort of like to thank the more general WashU community I've met on a whole, both through my class in DBBS and then the other years in my DBBS class in biochemistry and biophysics. We've all been a great group of people to just come up through in classes and to work with. It's been a very large sense of camaraderie among everyone, and that definitely makes doing research a little bit easier. And I also want to thank the cell bio department, people in the cell bio department, which is represented by just a picture of my lab. I don't have pictures of you guys. Barely have pictures of my lab, but I've really enjoyed working in this department. I enjoy how sharp everyone's minds are and how many resources there are and how many people there are to answer your questions, and just also how much fun it is to have a beer with you guys at Happy Hour on Friday and just unwind. So for that, I'm thankful. And then grad school is really hard, and it's made just a little bit easier by having some good friends. So I'd like to especially thank my group of friends that I made at WashU. Joe, I'm not going to call you Joe, Haley, Dan, Kara, and Kevin. This is us as graduate school babies, and then this is us as half doctors and half disgruntled elder graduate students. Anyways, my favorite times here have always been with you guys, so thank you for that. And then I also want to give a quick shout out to my two best friends from the earlier stages of my life, Elise, my college roommate, and Sarah, who has been my best friend since we were 4 years old. Neither one of them understand anything I do, but they support me blindly, which is a real skill and something that I treasure. And I also need to thank my family. Like Ken said, I was fortunate enough to go to grad school in my hometown. So I've gotten to see a lot of these people over the years, and that's definitely been helpful for this process. My extended family has all been always very supportive throughout my entire educational stages. And they've always been, especially the past 7 years, they've been super inquisitive in what I'm doing. So even though they don't understand it all either, they definitely care, which I appreciate. And then I especially have to thank my parents for, they've done a ton to get me to this stage in my life, and none of that would have happened without them. So I'll thank them for that as well. Now I will happily take any questions that you guys have.
So does the R7 heterotrimer, you showed it either bound to G alpha 13 or G alpha O? Do you know if it can bind both simultaneously?
I don't. I'm actually interested. That's one of the things I'm interested to see. Because if it could bind both simultaneously, that would provide at least a little bit of evidence suggesting that these things could be a point of crosstalk, immediate crosstalk between those different signaling cascades. So I think that would be interesting, and it's definitely something I've thought about.
So if they could both bind at the same time, what would that mean for pertussis toxin result?
Right. Okay. So for the pertussis toxin result, the pertussis toxin result could basically only informs me that there's not much basal G-alpha IO activity. But you can imagine a situation where G-alpha IO has been activated, like via cannabinoid receptors. And it's when G-alpha IO, R7 heterotrimers will only interact with G-alpha IO when it's active. So it could interact with G-alpha IO when it's active, when we actually add in a ligand under some condition, and also potentially be interacting with G-alpha 13. And the net result of negatively regulating G-alpha IO and positively regulating G-alpha 13, either under a basal state or maybe with a different activation other than LPA, would be cell rounding. So it could have a dual arm mechanism there. Still. Questions? All right.
Okay, if there are no other questions, thanks Stephanie again. And I'd like to invite you all back here at 4 o'clock. There'll be a celebration after Stephanie has been interrogated by her committee.
Thanks. It's the breakthrough videotape. It won't stop until the top.